So we have Eric. Welcome, Eric. We have that. Eric, if you didn't know, is our sponsor for this project. I'm sure that's surprising to many of you. Hey, Eric.
Hey, everybody. Great to see you.
And we also have William Eden, who is our resident sage and who did a lot of work with Eric, as a precursor to the project that we're taking on as part of the Portal Discord server. Hey, William.
Hey.
So let's see if Chad is here.
Yeah, good to go.
All right, folks, so we have something special for you. It's definitely still in draft form. Chad has been working day and night for exactly 1 day on this. So it's a little rougher on the edges, and nobody's seen it but him. So enjoy.
The World Health Organization recently put out a report, 32-page report, saying that aging is one of the biggest problems of our generation. Unless we do something to keep the elderly healthy and productive, the cost is going to crush national infrastructures, and our way of life, our economies are going to fall. And this is what the governments are saying.
We start losing brain mass at like the age of 20, and by the time, if you can actually make it to 100 years old, you lose like 20% of your brain mass.
The world's most important problem, namely the fact that we get sick when we were born a long time ago, and then we die, and that's kind of like not a good thing.
So the longevity function can, I think, be simplified into two components or two vectors. The first is lifespan. The second is health span. Can you reprogram progenitor cells? Can you selectively turn off senescent cells? Will, perfectly selective rapalogs come along? These sorts of things.
Ancestral wisdom and modern science to enhance longevity.
As long as we've been telling stories around campfires, our species has fantasized about breaking out of the cycle of mortality.
Aging is the buildup of air. That's all aging is.
Imagine a design space for living organisms. You can see all of the most extreme points to define the complete possibility space already explored by evolution. Then you can use technology and interventions to extend that design space into new points, effectively removing certain evolutionary trade-offs. The question is whether human biological immortality is contained within the convex hull of nature's experimentation and humans' innovation. We have a limited amount of time alive. If we lived longer, we would be able to experience life for longer, have more time to work on projects, experience progress, and share our knowledge.
You think about the investments required to learn music, computer programming, science, like all sorts of things you might want to integrate. Almost nobody's going to get to do them during a life.
It seems another great benefit is the ability to coordinate and think about long-term problems, as our range of thinking seems proportional to our lifespan. Here are some of the goals of the project. Investigate the relationship between nature and longevity. Next, look into how we might leverage technology to indefinitely extend biological constraints. Explore why radical longevity with healthy lifespan is necessary and timely to pursue. Lastly, apply our research to benefit society through scientific discovery and cultural progress.
This is the Convex Hull project of radical longevity.
So that's kind of a quick introduction to the Convex Hull project. We've been working on this for a number of weeks. We are not yet really even halfway through. What we've been spending our time on is researching biodiversity in nature. And so we're going to go through some of our research really briefly. Before that, I want to give Data the floor, who's going to talk a little bit about how we're structuring the project. We're currently keeping everything in a single Google Doc. It's currently 81 pages and quite unwieldy. So Data has spent some time thinking about how might we use Google Drive, split up the docs, make it easier to consume, easier to contribute to, and easier to get others involved in.
All right, so thank you very much for your time and for having us. First of all, I'd like to talk about our project. As the video explained what it is, I want to go into what is the structure of it and what exactly are we trying to solve and break down. So I created this process that's on the stream using a combination of this engineering method, the scientific method, and descriptive writing techniques from my middle school. And I wanted to address some of the essential questions. For instance, when you start with a project that you want to improve on, the first thing to do is define the project so you know exactly what's going on. So I've split it into category 1 is defining the project. Category 2 would be background research, evaluate why the project is viable and tenable, and assess possible paths to solutions. Something that's important is truth. If you would scroll down to possible paths to solutions, which is in 2B. What I've done is I've started to create a structure. I wanted to base our project of longevity on the human life cycle, right? And I've broken it into a couple different places, right? Like, for instance, one part of our life cycle is reproduction, one part is our lifespan itself. One part is the reversing the life cycle, right? What is regeneration? Changing the life cycle, our genetics, our environment, in order to change the world in which we exist. And pausing life cycle as well, although we haven't looked into that. And I think that breaking our project down into these kinds of base paths of solutions is essential to understanding what part of it we want to assess, because this question, like so many, can rabbit hole in a million different ways. And so understanding scope and scale is imperative when trying to learn more. We also wanted to understand to understand the problem, we not only have to understand what life is, but what death is. So additionally, I've created a couple extra pages towards the top of this document, which I can post publicly, that talk about what does it mean to not be alive anymore, whether it's external reasons, base resources that have not been met, or internal reasons such as running out of there's a thing about myelinization and that whole issue there. So creating these paths is going to help us to narrow our research as well as pick where we want to focus. That being said, this document is hopefully going to serve as a base outline, as well as help us create a place in which to categorize all our research and act as a central hub of information so you can easily find what you're looking for. In the bottom of the document also are some alternatives, as well as specifying what the research means and is. So for instance, how would we apply this pragmatically, and what are our constraints, because these things are also important to look at scope and scale. That's about it for the Project Center.
Thank you so much, Data, for going through kind of the doc we had, abstracting out a sensible structure, and then putting it in a form that everyone can use. So there is a Google Drive, which we'll make available to contributors and to anyone to view. And that's where kind of the finalized work will go in. And then we have our main project collaboration document here. I'm not going to go through this, but you can see that we're documenting our action items, to-dos, the team. Here's the team. Shout out to the team. Ton of really great work being done. We would love for anyone to join us and contribute to the project. We've got an abstract, and then what we have is like a rough outline that we're working on. And Teisenberg's going to share with us a little bit about kind of the metagame that we're thinking of here, and why radical longevity might be related to our culture and our civilization at like a broader level. So Teisenberg, do you want to take it away and give us like a 3-minute summary of what you got here?
Yeah, sure. So I think for as interesting as the radical process, we should give a lot of thought as to how we're going to integrate it and how we our lives and our. So the first thing is it has a capacity to be tremendously disruptive. With rivalrous games and exponential tech leading to self-termination. For a deeper discussion of this issue, I would refer you guys to the Portal episode with Daniel Schmachtenberger. Pretty much with exponentially increasing capacity and availability of destructive power without an equal or greater increase in wisdom or implementation of non-rivalrous dynamics, the human experiment will result in self-termination. We are apes with laser beams. This is as certain as any formula that you can guys really find. So it's really not naturally adaptive. We didn't evolve to live beyond our current lifespans. So we're not really psychologically equipped to handle it gracefully. We all have people that we love and care about that we do not want to die. So with that prospect on the horizon to stop that, I think it's valuable to think if we would all really handle it in a mature, responsible manner. So there's just challenges on that side of it. So it's no less dangerous than any of the other advances in tech, with the other lack of wisdom and instincts that we have. So if we do aim to successfully challenge this, then the advance in wisdom must be paired with it, which it has to be paired with everything else. So that should definitely be a focus. So the empiricism without epistemic humility leads society to a lack of coherent teleology. So this must be paired with a purpose beyond our simple desire to have it. Farming addresses our desire for food, social media addresses our desire for communication, but are we really utilizing our abilities in these areas correctly or responsibly? I don't necessarily think so. Radical longevity might address our desire to live longer, but it won't improve life in and of itself. Beyond living more, we also have to live better. So I personally think this could help orient us towards higher purpose goals. The situation is we are as gods, but we don't feel like it. We die like any other animal. So I think that radical longevity provides the opportunity to really make us rethink our capability and the reality of the powers that we have at hand. I think that there's a way that we shirk higher responsibility as goals of humanity, just because, moving to other solar systems, exploring the stars doesn't seem feasible within our current lifespans. But if the technology happened where we could live for hundreds of years, suddenly that seems more in the purview horizon. So the metastability. So as I said, it must be delicately implemented. So if not, what could possibly be more destabilizing than discovering the key to immortality? So this has the potential to really exacerbate everything not handled properly. So two possibilities: everyone gets radical longevity. What now? I mean, all the practical considerations that come with that. Where do we fit everybody, and all the other things? What does this do to our relationships, consciousness, memory, neurological function? Every other assumption that we all are currently making in our lives being mortal, what happens when that changes? Or some people get radical longevity. Who gets it? Who decides? What's the process of who gets it? And if the inequality of income destabilizes society, the inequality of lifespans will be worse if not sufficiently addressed, as one can imagine. So with the obvious population boom that would come with radical longevity, it must be paired with sustainability to not burn out resources at the rate that we are at currently. And much like wisdom must drastically increase to offset our exponential increase in destructive tech, sustainability must drastically increase to offset radical longevity. So maybe we should start a radical sustainability conversation to accompany this. As far as humans, apex predator, niche exploiter in the natural environment, as I kind of just referred to, radical longevity will just enhance our impact on every niche we've already exploited on the planet. And niche within the social context, those who are most adept at gaining asymmetric power do so. So naturally, those who would most want to control radical longevity technology for their own means to exploit themselves will also attempt to do so as well. So just another thing to keep in mind. And I believe that is it for me.
Excellent. Thanks, Teisenberg. So we did a bunch of research on biodiversity. Eric seeded us with a lot of really interesting animals and strategies in Mother Nature to investigate. Here's a list of them. We would love contributors. Please come help out. By the way, for the video that you saw earlier, Chad could really use some help picking out really great quotes from YouTube videos and podcasts, finding assets of like seahorses, mitochondria that are really cool, either animations, videos, or audio, or still images. So if you'd like to contribute, please join us in the Convex Whole chat room. So we're going to go spend a couple minutes and for each of the strategies that our contributors have worked on. And yeah, we just want to get kind of one insight from each of the animals that we did research on. We're going to turn these into consumable videos and stills and other stuff later. But each of us did, took a couple of these and did like a 2-page summary, and we'd love to share that with you. Actually, I just realized, Simon, we wanted to go through telomeres. Why do we care about telomeres? So, Simon, are you on the stream? Can you take it away?
Yeah. So somehow I ended up with this topic. It's scary, given the attention that Eric and Brett, of course, have brought to the topic. So bear with me. What I wanted to highlight are not the topics that Eric and Brett have covered in much detail yet. Rather, taking a slightly more pragmatic view or concrete view of the problem. So what I did was go over some brief examples of why we care about telomeres, what they do, what they're for, then address some of the problems and intractabilities of telomeres, and then where do we go from here? So what are telomeres? Well, you'll all be aware that they are basically non-coding sequences that cap chromosomes, the integrity of the coding and regulatory sequences within the chromosomes. The neat thing about them is that with each cell division, they shorten so that each of the child cells have a shorter telomere than their parents. That places a limit on the number of divisions that a cell lineage can undergo, and that is hypothesized to protect against cancer, for instance. These processes basically raise a few hypotheses and predictions. I mentioned the idea that this limit in the number of divisions, which is called the Hayflick limit, is a protection against uncontrolled replication, i.e. cancer. Telomere length can potentially be used as a measure of the age of organisms, and specifically the biological age of organisms. Therefore, the rate of telomere loss might predict longevity. So seeing how fast your telomeres are shortening might give an indication of your age at death. Telomere shortening factors such as infections and environmental stresses, which have been demonstrated in the literature, are likely to shorten lifespan. However, there's a number of problems with telomere in practice, and using telomeres as a metric of age. There's a huge amount of variability between tissues within the same organism, between individuals within the same population, So it isn't very clear what the actual length of a telomere at a given time tells us. There's another problem, is that when you measure length in different species, you can find tissues in which telomeres are elongating rather than shortening over time. And that's a problem that's been observed in lab mice, and to belabor the point of what lab mice can and cannot tell us. There is an implicit assumption as well that telomeres stand in somewhat of a causal relationship with biological age, that if you lose all your telomeres, then your tissue is basically old and you're probably going to die of it, but it's not very clear whether they are in fact only a marker of age or whether a cause of biological aging. So, given those problems and the fact that they are actually intractable or not always tractable on a practical level for us, where do we go? So one way to address these unresolved problems with telomere lengths would be to do some experimentation in some way and a comparative approach. So much like the comparison between the lab mice and the wild mice that Eric and Brett have explored, but extend that to more species and more environments. Another way might be to find better clocks than telomeres. And currently there's a huge interest in DNA methylation, and there's a much tighter correlation between the level of methylation in the DNA and the actual both chronological and biological age of organisms. And the idea there would be, for each organism, you can measure chronological age in some tissues and biological age in the tissues that are undergoing more stress, say. And that delta might be more predictive of your likelihood of dying. Another angle that I discussed with data actually yesterday, and I wanted to mention here, was given the imperfection of these various measures of age, can we still do something about this? And maybe one way forward would be to actually propose that individuals draw a profile of the telomere length at a very young age, and then regularly get an update on the telomere length for each tissue of interest. That would give an individual-based rate of telomere loss. It might be telomere, it might be DNA methylation. The logic is the same. Basically, you get a clock on each of your tissues. And the idea there is then to intervene on the tissues that are aging the fastest. So if your skin cells, for instance, are aging faster than they should otherwise, then you should adopt behaviors that will protect your skin from external stresses. I'll stop there. Happy to take any questions later.
Excellent. Thanks, Simon. Bristlecomb pines. Richard.
All right. To quickly go through a few different plant species, one being a non-clonal organism and the other clonal. One of the issues I noticed from the literature regarding longevity in plants is that a lot of the information is focused on those things which are geared toward human life extension. So there may be some things missed when investigating plants for plants' sake rather than plants for humans' sake. With that in mind, bristlecomb pines are the oldest known sexual reproducing non-clonal organism. They're unique to other long-lived trees, and there are many others, in their slowed growth rate and the extreme conditions in which they live, which appear to select for dormancy. The interest this might have for this project is the ways in which decreases in metabolic activity can maintain a stability within a life form and slow the metabolism and increase longevity. The implications for this might be important considering the need to leave this planet possibly, or even the solar system, and how human beings, if they wish to stay in a biological form, might do so. One of the ways in which they do this relates very much to what Simon was talking about, is regarding telomere length and telomerase activity, which is an enzyme which can help repair some of those non-coding regions after they've been damaged from radiation or cell reproduction. The interesting thing in particular about plants is that they have two main points of growth and stem cell locations, one being underground. So they have obviously their, where you see the root tips, maybe at this time of year, you see things blooming around your home. You see the apical meristem where you have lots of growth. You also have lots of growth at the root stem. And that provides an area where you have stem cells and cells that are multiplying quickly, yet they are being protected from things like UV damage, and this is a possible reason why they're able to maintain robust and intact DNA sequence while existing for up to 5,000, almost 600,000 years. And those are the two interesting applications for human beings. Moving to the clonal organisms, there are many clonal organism species of plants, and the numbers regarding their age range from tens of thousands of years to hundreds of thousands of years. Either way, I found the application to this project more cultural and philosophical, looking at what it means to be an organism, what it means to have a continuous life. A lot of conversations about human longevity involve an individual as a person. We might be able to question that idea as a distinct biological entity. And if we look at a larger collective identity, which we cannot necessarily implement from biology, but something that might be culturally relevant. We could use tools in which we see ourselves as more of a collective organism, and that version of cloning rather than just the actual biological cloning might be a useful mechanism for extending the lifetime not only of individual humans, but all humans, especially if we see ourselves as a species, as a unitary entity, then maybe that long-term vision is able to be enhanced. that is all for now.
Excellent. Thank you. Timmy, with the seagrass meadows.
The seagrass, Posidonia oceanica, it's probably one of the most extreme examples of a clonal organism living for a really long. This one giant meadow southeast of Spain might be as much as 100,000 years old. So there's a lot of different factors that contributes to that. Kind of when you're thinking of clonal organisms, essentially able to grow forever. So while regular organisms eventually cap out, they don't have to really worry about that. And so telomerase production can pretty much continue through asexual reproduction and stuff like that. And it's kind of really important too that it grows slowly. So this specific seagrass that I was studying, I think it grows at like centimeter to 6 centimeters every year. It's not really that ridiculous to think of how extremely long times, because you kind of have that slow growth. But another kind of really important thing about this specific seagrass is you can kind of imagine that a clonal organism is sort of starts from one center point and grows out, and each of those branches sort of like their own little cell line. But a lot of clonal organisms, what they can do is those different branches can go back and reconnect, and when it reconnects, it will eventually resolve any mutations, any DNA that might have been lost during growth outwards. So this can't really be used in humans. You know, you can't have humans growing kilometers in length. Goes back and reconnects and fixes its DNA. Well, I mean,
Extreme.
it's like sort of just
we're getting some feedback.
Would maybe taking blood or at a young age and reintroducing them to something, and possibly taking parts of a young person's body and introducing them to their elders. And I think actually a few companies have actually tried stuff like that with limited success, but yeah, that's pretty much it.
Cool. Thanks, Timmy. Transtar with the beautiful naked mole rat.
All right. So, like you were saying, the lovely, beautiful naked mole rat. I've kind of been following this mammal for a little bit of a long time. It's kind of been in and out of popular science articles and everything else, but I've tried to keep as many tabs on it as I could. So the one thing with this is it tends to have actually bucked the trend on the size equals the span of life, whereas, larger mammals usually tend to live longer, smaller mammals usually tend to live shorter. If you think about the lab rat, that usually lives 3 to 5 years. These naked mole rats, they are about the same size. However, they live almost close to 30 years. So it is a very stark difference. The big thing with the naked mole rats is they do live in an environment in West Africa that is very harsh, has a lot of oxidative stress on them. So by all accounts, they should be developing cancers left and right. However, they seem to be cancer-free. They do not seem to develop cancers even when people who keep these in the lab try to introduce cancers into their systems. It just doesn't go anywhere. Going down into their cellular functions, it seems that they have, both by behavior and by the cellular functions, exceeded at protecting their DNA and making sure that there's no mutations, no adaptations that kind of take hold of their DNA. I say behavior-wise because they are a colony species where there is essentially a queen naked mole rat, as lovely as that sounds, and she is the one in charge of her colony. She's the only one allowed to reproduce. There is competition for her spot. However, this vastly narrows down the amount of genetic variability that you do have. Meanwhile, on a cellular function, their telomeres are actually very similar to the length of humans. There are a couple studies that were like, oh, it's much shorter than the lab rats and lab mice, and many people will find that funny. But the telomeres do not seem to die off as quickly as one would expect. In addition to being cancer-free, also their lifespans are very long as well, whereas they can hold reproductive, or they can reproduce well into almost the end of life. So within 1 or 2 years, they're still able to reproduce and show no signs of aging as well. Their cells are very good at protecting their DNA via the proteins and the DNA repair mechanisms. A lot of that has been speculated to be how it's the DNA's topology proteins are, but there is still much research being done on that at the moment.
Excellent. Thanks for sharing. Nathan, are you there for sponges and germline immortality?
Okay, this will probably be short and sweet. I didn't do have a lot of research and to get much in depth, but I think from the perspective of simplicity, I think sponges should be looked at. There's not a specific timeline on how long they can live, but some can live probably up to 11,000 years. And I think based on the fact that they don't have nervous, digestive, or circulatory systems, kind of gives them extremely good assets to be able to live a long time. That could be some of their benefits. They're mostly sessile, which could definitely be put under some cost issues. So, in some relevance to human biology, they use a similar complex gene regulation toolkit, similar to what humans do. Some other properties, their ability to suffer damage and regenerative capacity is a positive.
Sweet. Do you want to tell us about progeria? Or no, sorry, germline immortality?
Yeah, this was probably my favorite part to research, and it's the least I know about among the two. Sponges were most interesting because of how simple they were and how long they can live, up to 11,000 years, and there's different research. But for germline immortality, I think based on what I researched and my current knowledge, I think looking at the hereditary cell lineage and if it's possible to have immortality through the germline, based on my research, I'm not necessarily sure if it's proven. There is some research out that, based on the wording, kind of says that there can be some germline immortality based on primordial germ cells, and so the longevity might have something to do with signaling between cells with certain genes. And so it might be helpful to go back into what I put under the research and look at human germline engineering and see what that might have to do with effects on germline immortality, and then also what CRISPR might be able to do to take a better look at that and see how editing would either prevent or enhance longevity. I don't have much more than that on human germline or germline immortality, but based on what I know, that's kind of how I approached it.
Thanks for giving us that summary.
I will give a quick synopsis of the lobster and the Greenland shark. The reason why telomeres are important is because while researching lobsters, I didn't find any info on their actual telomere length, but they do. Their primary method of longevity is that they produce telomerase throughout their lifespan, which replenishes their telomere length to whatever length it is. So lobsters essentially have biological youth throughout their entire lifespan, and the longer they live, the more virile and hardy they become. But at some point, they just get too large from constant growth, and their metabolic requirements become too great. Because of that, 10 to 15% of lobsters die during molting, just because they're so huge, and they essentially die of exhaustion trying to crawl out of their giant shells. And if they fail to molt, then their underlayer becomes exposed bit by bit, and they essentially lack armor and end up dying to the elements. So that's how lobsters, their strategy works. Probably not a great strategy
All right.
For human forever. Yeah, and then the Greenland shark, all sharks actually have, at least all sharks that I've looked into, have a large number of redundant DNA repair proteins, which a lot of animals, I think they have like 10 times as many as we do. We have like 2 or 3. And then, as I said, they have redundant copies of them, so they have a ton of them. But the Greenland shark in particular, it lives in near frozen waters, and it has somewhat toxic flesh, so it doesn't really have predatory concerns, and because it's in such a cold environment, its cellular metabolic rate is drastically lowered. Their active organism metabolic rate is also fairly low. In fact, they're known as sleeper sharks because they essentially just float in a direction, not responding to any outside stimuli throughout. They don't even reach sex about 150 years. They live 500 to 300 years, and they essentially just have such a low metabolic rate. There's not a lot of wear and tear in their DNA from, they don't have to constantly be replacing cells. It's similar to the mechanism where when you near starve lab rats and lower their metabolic rate, that they live 10 to 15% longer. This is essentially an extreme version of that, which synergizes with the environment they live in.
They look pretty cool too. Look like they could go to war.
Essentially, this field is so wide that you have what is similar to a timeline within, I hate to say it, but maybe quote-unquote infinities on either end, and, infinities in between, which means that every single question has so many permutations, and, why is this? Well, why is this? Well, why is this? Into these rabbit hole chains, that it's incredibly important to understand scope and viability. And these two things, scope and viability, I want to address in the next stage of that document, which was narrowing down our field of interest based on our background research, as well as picking what we think to be the optimal, path to take, or splitting into groups for different paths to take. Particularly, my research was I decided that nutrition was going to be the path I wanted to take, so focusing on the current life cycle. And in nutrition, I found a strong correlation between nitrous oxide and the immune system and the inflammatory response within the immune system. And something like, and then the solution to that would be found in things like agarikon, and you can go ahead and read that on the paper that I'm going to go ahead and post in the general chat. But basically, the main takeaways are scope and scale, and narrowing the field of research to avoid rabbit holes.
Josh, do you have a few minutes you want to go through the biohacking stuff?
Yes, I can. So the section that used to be called biohacking is now kind of called maximizing health span and lifespan, and it's kind of going into the relationship between health and so I've broken up kind of optimizing health span and lifespan across 4 vectors. We've got maintenance, which is everything from intermittent fasting to sleeping enough to making sure you get your vitamins, proper nutrition, hydration, all that sort of stuff. And then there's hacking, which is everything you can do without the use of kind of medical or other avenues. So things like stress management, meditation, sauna and ice bath use, supplementation and extended fasting. And then things like medical therapies, so microbiome transplants in general, medication, things for diabetes, does have some impact on longevity, just anything you can do to try to kind of fix issues in your body. And then enhancement, which is kind of the else, which is what a lot of the rest of the Convex Hull project will kind of target, which is things like implants, brain-computer interfaces, nanorobots, genetic engineering, stem cell use, tissue engineering, all that sort of stuff. And so what we're doing right now is just working on fleshing out the list of all these 4 different vectors and getting all the information there and providing clear and easy paths that people can take to start to improve their health. Yeah, I think that's it.
Excellent. Go ahead, Avicard.
Okay, great. So I looked into Hydra, and Hydra are rather simple. They're based a small genus of the phylum Cnidaria, class Hydrozoa. They're usually native to temperate and tropical regions. there's been a lot of interest generated about the regenerative capabilities of Hydra. And specifically, there's some very good research to suggest that Hydra actually don't have senescence, which is very interesting because they have a very high, even though the exact telomeres in Hydra isn't exact understood yet, there's some anecdotal evidence that they do have telomerase, although that's still being looked into. What makes Hydra very interesting is that they have their regeneration absolutely incredible, meaning there's been stories of, there's some reports of people putting Hydra in a blender and then it reforming. If you cut a Hydra in half, it'll turn into two different Hydra, and the different with different gradients of morphogen in the that allow it to differentiate into a head and foot group that the Hydra usually reproduce asexually, but there have been reports of Hydra, although sometimes some species will reproduce sexually and then die, which would point to something special about the asexual reproduction. The Hydra, they have 3 different bodies of stem cells, but somehow manage to avoid the senescence could come longer. I haven't defined whether they are cancer. It seems that they are to some degree. But what I found absolutely very interesting with them is that the special properties of the Hydra seem to be linked to the FOXO gene, which is a gene that is that's being studied in humans now with regards to apoptosis, cell death, which is programmed cell death, and the regulation of the cell cycle, meaning that it's it's excellent at stopping. So it would be, I thought the interesting takeaway here was that it's the combination of the stem cells and FOXO, which would be a very valuable tool for stopping cancer in a species that has such a constant turnover of stem cells and the ability for it to constantly be regenerate into new organisms as well. The implication for humans would be some kind of therapy or some kind of research that would look at not only the FOXO gene, its application stopping cancer with rapid stem cell use and a lot of the other therapies being played around.
Cool. Thanks, Avakar. The Hydra is super interesting. It is like less than a millimeter in size, so I don't know how much we can learn and apply to humans unless we want to shrink ourselves. But
right. Well, they're incredibly simple.
So that's our progress. This is the Convex Hull of Longevity project. If you'd like to contribute or keep tabs on where we are, go to the chat room, #Convex Hull of Longevity. Please contact me if you want to contribute. What we're looking for immediately is anyone who wants to do research on some of these biodiversity strategies, anyone who wants to look into finding great quotes about longevity from thought leaders, or pulling resources from the internet, like images that are really cool about each of these animals. Until next time, thanks so much for joining us, and we will kick it off to discussion. Thanks so much to the team. We'll catch you on the next one.